So what you have after library prep is this, a set of DNA fragments with a blue adapter on one side and a red adapter on the other side of the. What you have on your flow cell is this. So on the surface of the flow cell, there are two different primers covalently attached to the surface of the flow cell. Why two primers? You have one for each adapter. So the green primer will bind to the blue adapter and the purple primer will bind to the red adapter. You take your library, you do a denaturation, so all your double-stranded fragments will become single-stranded, and you put your library on the flow cell. So what will happen? The blue adapters will bind to the green primers, red adapters will bind to purple primers. So your fragments from your library are attached via this primer binding to the flow cell. Now, this is still a very loose attachment. You cannot work with this. What you need for sequencing is a covalent attachment, so a very strong attachment. So this is where the next step comes in. So what you do is you use the green primer as a PCR primer. So you're going to do PCR, and by doing PCR, you're generating a double-stranded fragment again. The original strand from the library is here, and its reverse complement is now covalently attached to the surface of the flow cell because it's covalently attached to the green primer. So now you have a strong attachment to the flow cell. So in the next step, you're going to do a denaturation again, and the original strand is washed away. Before you can do sequencing, you have to do an amplification first. You have to generate enough copies on the surface of the flow cell to generate a signal that can be picked up. So how is this done? They make the conditions on the flow cell such a way that these fragments can easily bend over the surface of the flow cell. So they can't go away because they're covalently attached, but they can bend to all sides. So at some point in these conditions, the other adapter, the red adapter at the other end of the fragment, will find a purple primer on the surface of the flow cell, and it will bind to that. And this is what is called formation of a bridge. You see it has a bridge-like structure. Now, the purple primer will be used for PCR amplification. So you will get this double-stranded fragment, but now both ends are covalently attached to the surface of the flow cell. So you do a denaturation again, and you repeat that process again and again and again and again. And after you have repeated that process thousands of times, you will get this on the surface of the flow cell. So these are called clusters, and you will have millions of clusters on the surface of the flow cell. So every cluster on the surface of the flow cell, this is one cluster, this is another cluster, every cluster originates from one fragment of the library. Yeah? So this here, what I made here, represents one fragment from your library and its reverse complement. So you have this one and its reverse complement. It was made by PCR. And you're going to repeat that thousands of times. And the ultimate goal here is to make clusters in such a way that they contain thousands of copies of one single fragment from your library and its reverse complement. Now, since you have no control over where the clusters form, it is of course possible that you will sometimes have overlapping clusters. So clusters that represent more than one fragment of your library. So you should avoid that as much as you can. Yeah? They don't put a lot of fragments on a lane just to make sure that they are distributed far away from each other so they will not start overlapping. Because overlapping clusters is really bad. So if you have a really big library and it's too much to put on one lane, then you just have to simply put it over multiple lanes. So this is the general principle for the flow cells that consists of glass slides. So as used by most sequencers of Illumina. Illumina has a few sequences that work with a different principle, and I will come back on that.